Ms. Doyle Bose-Pandey from Indore. Madam is a course director and trainer at Embryology, an academy for learning hands-on embryology at Indore. Her chief areas of interest are oocyte vitrification, ICSI, embryo pooling for poor responders, vitrification, and warming of gametes. Currently, Madam is working at Indore Infertility Clinic as embryologist. She is also a clinic manager, managing all operations pertaining to an independent IVF clinic setup. Her love for learning new things made her master embryology in a short while. She has trained many embryologists over the past and believes in making IVF treatment at affordable for all. We welcome you, ma'am, and her topic for the session is on embryo culture environment, mimicking nature in IVF lab. We welcome you, ma'am. Kindly take over the session. Any queries or questions can be posted in the chat box. Kindly all participants to ensure that audio is muted.
Okay. Am I audible?
Yes, ma'am. Yes, ma'am.
And my screen is also visible?
Yes, ma'am.
Okay. First of all, a very warm welcome to all of the delegates, and I'm very happy to see such a participation on just for academic interest. And thank you, Dr. Mini, for such a kind introduction. I feel very important with your introduction. So, my experience in embryology has been nearly 6 years, which is minuscule when you compare with most of my counterparts. However, my learning experience has been very recent, so I exactly know how anybody who's new to embryology feels when how overwhelming it could be when you are studying embryology and trying to work in an IVF lab. So I hope that with this topic, which is very much a fundamental topic of embryology, and I hope I have tried to gather all the information in a presentation. And with this presentation, I hope most of you have more clarity than confusion. And if you have any confusion, through the Q&A session, hopefully I'll be able to solve that. So without wasting time, let's get going with the presentation. And let's see what we all will be covering today. So even before we try to understand what we need to do in an IVF lab in terms of an embryo culture, we have to first understand what happens within our body, what happens in vivo, and what is the journey that an oocyte or an ovum, right from the time that it has been released till the time it is fertilized, and when it passes through the oviduct and it reaches the uterus for implantation. To know this entire journey and what are the different environmental factors that the embryo faces during this journey. And we will try to mimic the exact same thing within our IVF lab, and hopefully if we are close to what happens in vivo, we should be successful with good quality blastocysts, which will eventually lead to a pregnancy and a live birth. Now, the second thing that we need to understand is what are the different components in an embryo culture system. The culture medium, which is again, if not everything of embryo culture system, it is definitely a central part of the embryo culture system. To understand what are the different embryo culture protocols and strategies which are available, and some basics about culture condition, and some take-home message. Now, with this, okay, I think somebody needs to mute their mic. Okay. Now let's see what happens, what kind of environment an embryo faces in vivo. So in vivo, the in vitro culture simply tries to mimic this environment. And Firstly, we see that we have balanced nutrients. So there are a few nanoliter of oviductal fluid which is available to a fertilized oocyte from the time it is being fertilized till the time it is implanted. And this balanced nutrient is being tried to replace that with the embryo culture formulation. Then we have an optimum temperature, humidity, pH, and osmolality that the oviduct provides to this initial compaction and post-compaction embryo. And all of this we try to achieve through a culture media, a buffering system for maintaining pH, and a robust incubator to give you that kind of a humidity, temperature, and concentration of different gases. The third thing which is important in our in vivo system is the optimum amount of oxygen and CO2 tension. And this we try to achieve through incubators. Have, uh, our body prevents from external contaminants and oxidative stress. And within, we try to constant motion helps the embryo. And this we try to achieve through embryo culture protocols, culture strategy, and an embryo culture platform. There are different kind of suffixes which have been used to the word culture, like platform, system, protocol. I hope with passing slides, we are able to concentrate on one thing and understand what a platform means, what embryo culture system means, and all. And if there is any doubt, we'll definitely solve at the end. Now, trying to elaborate the journey of an oocyte from the time it is being fertilized till the time it reaches our uterine wall, as we see, we can divide this journey into two parts: a pre-compaction embryo, which is day 0 kind of, and a post-compaction embryo. It's after day 4 till the time it reaches the uterus and implants somewhere around day 5, 6. So there are stark differences between the environment, the immediate environment of the embryo, the pre-compaction and a post-compaction. So pre-compaction, the embryos are in control of maternal genome, and post-compaction, the activation of embryonic genome occurs. Now, what is maternal genome and what is embryonic genome? So these are like buzzwords, right? So I'm not going to get into details of genomics because that is a very detailed and thorough topic. But I think to just put in simplified analogy, just like a mother handholds her children when they are born to teach them how to eat, drink, clean, and so on. And then at the right moment when she feels that the child will be able to do these things on their own, she just leaves them to do this under her supervision, and then finally eventually leaves it completely. So maternal genome is kind of pretty much the same thing. Here, um, the gene products of the maternal, the mRNA and all, they regulate the initial meiosis, fertilization, and the early development of the embryo. It is kind of hypothesized that it is the maternal genome which indicates and the switching over from the utilization of the maternal RNA to the embryonic genome occurs, and this is triggered, this is controlled by the maternal genome itself. So this is called the zygotic genome activation. Even if you don't understand too much of the details of maternal and activation of embryonic genome, I think what is most important to understand here is pre-compaction embryos are very susceptible to their immediate environment. So they are very susceptible to minor changes in pH, temperature, and osmolality. While as the post-compaction embryos are much more robust because of the number of cells that they have, and they can regulate temperature, pH, and osmolality much better than the pre-compaction embryos. And hence, pre-compaction stage, the best is them untouched as far as possible. So don't just fiddle around things. Then be very gentle with your gametes and leave them as much untouched as possible. Other major difference is the energy source or the energy substrate. The initial energy source during the pre-compaction stage, the main source of energy is pyruvate and lactate. It's not glucose. While as in post-compaction stage, the source of energy or the energy substrate is. That's why the metabolism initially is called an oxidative metabolism, which is more quiescent, and the metabolism in the post-compaction stage is a glycolic metabolism. The third difference, which is a major difference, is how non-essential amino acids are used in the pre-compaction stage, and both essential as well as non-essential amino acids are needed in the post-compaction stage. So now you guys know probably amino acids better than me, but there are around 20 or 21 amino acids, and non-essential amino acids are the one which your body cannot synthesize. So sorry, essential amino acids are the ones which you're, let me just, yeah, are the ones that you cannot synthesize on your own. So the cells initially will not want to be dependent on something that you cannot synthesize. So initially, you will want to be dependent on things which are available to you readily, right? So this is the major difference between a pre-compaction and post-compaction, and also you have differences in the temperature, pH, and oxygen tension. Like shown in the slide, the temperature in the pre-compaction stage is about 35.5 to 36 degree, while in the post-compaction stage, the temperature which the embryos are subjected to are close to 37.2. So there is this kind of 1 degree variation in the temperature between the pre-compaction and the post-compaction stage. So at the site of fertilization, it's important to note that the temperature is slightly lower, which is 35.5 degrees. The pH also is different. The pH in the pre-compaction stage is 7.94, while that of the post-compaction stage is 7.2 to 7.3. While as we go forward in the slides, you will see that the pH is actually a high, low, high kind of a paradigm, which we'll see later on. And the oxygen tension in the pre-compaction stage is approximately 5%, while that of a post-compaction stage is approximately 2%. So let's move further. Now, embryo culture system. So everything which comes in close vicinity to our embryo is a part of the embryo culture system, so including us, the embryologists. So the central part of the, what do you understand by a culture system? There are various factors which come together to give an end result, and even slight or minor variations on one part is going to affect your development of the embryo. So these are the 8. I try to list a few things which are part of the very important part of the embryo culture system. Embryo culture media, naturally the centermost part. Then you have the kind of embryo culture dish that you use, the type and the quantity of oil that you overlay on your dishes, gas phase for equilibration of media, temperature and humidity, inhibitors that you use, air quality in your IVF lab, the embryologist himself or herself, and the type of gamete handling, how precise you are at your gamete handling. These are all part of your embryo culture system. Now, let's go quickly to embryo culture media. Now, today we'll try to cover what are the expectations from our embryo culture media. We know that the first and foremost expectation is that it should ensure growth of an embryo, to decrease the embryonic stress, and it should also maintain the gamete viability. These are the 3 foremost expectations that we have from our culture media. About the history of embryo culture media. If we, instead of going through each of this, I think it is important to note that we started as early as 1929, where first we tried to culture rabbit embryos in plasma. Then slowly and gradually over the next 50 years, we reached step 2 and Edward reached a point where they used a simple solution with pyruvate supplemented by patient serum. And this is in the environment in which the first successful test tube baby was cultured. Beyond 1978, a lot of things happened with the embryo culture medium, where Menezes and his friends tried to add a few serum albumin, and that the major thing that they noted was serum albumin resulted in non-adherence of the embryos or the gametes to the glass surface. 1990 onwards, we came to, after the advent of computers, the scientists took help of computer and something called a simplex algorithm for optimization. And they tried to identify and alter the concentration of components which at a higher concentration were detrimental to the embryos. And this gave rise to the medium called simplex optimized medium, or SOM. And this was the stepping, this is what gave rise to the single step media commercially. And after 1990, we see that two lineages of the culture medium. One was a sequential media group, which is called a back to nature, and the other was a single media group, or the one which followed through the SOM culture media. And this was the single step media, let the embryo choose. So we still have, both of these culture strategies, protocols are very different, and we still have a lot of debate on it. People who vouch for sequential, they prefer to go sequential way, and there are many people who prefer the simplified single step media. And both of them have, benefits and disadvantages of their own. So it is up to us how to choose based on some informed decisions. Now, instead of going through the composition of the culture media completely in detail, we won't be going through what are the purpose of amino acids and fatty acids and all, because we will be short of time. We have a lot of things to cover today. What is important to know that commercially, I think there are about 10 to 12 companies who formulate media, culture media. And this new generation culture media has more than 80 plus components in them. These components are majorly divided into these: water, basic salt, carbohydrates, amino acids, fatty acids, EDTA, vitamins and antioxidants, growth factors, antibiotics, etc. and etc. None of these companies, the commercial companies, will let you know the percentage, the exact constituents of the media. That's always a trademark. So, but they will tell you that they have, what is the salt that they have used, but maybe not the exact quantity of the salt. Key components of a modern medium. So if we saw that we have one media system is the single step, then sequential media has two cleaving media and the blastocyst media. So if we compare a single step media with a cleaving and a blastocyst media, we see that the energy substrate is kind of all of them have glucose, lactate, and pyruvate. However, the quantity of glucose, lactate, and pyruvate would be different for one media, would be different for a sequential and a blastocyst, cleavage and a blastocyst media. The cleavage media should have lesser amount of glucose as compared to the blastocyst media. When we talk about the non-essential and essential amino acids, we see that the one system has all the 8 non-essential amino acids, while the sequential cleavage media has 9 amino acids, and so on. This is just a chart mentioning you a few things. One thing which is important to notice: chelators. Chelators like EDTA. So like we see, EDTA is said to have a detrimental effect over plastulation, and that is why in the sequential media, you have no EDTA, while you have EDTA some, minimized form in the single step media. Vitamins, we have, again, this is one chart where it is showed that the sequential blastocyst media has 4 vitamins in it, while it is not present in the single step and the cleavage media. Purpose of vitamins, again, it has a lot of antioxidative properties. So this is just a charting. We don't need to learn this by heart. This is just, something which you should do in culture media when it comes, when you open the bottle. This is a kind of a chart that they will mention. And you can, usually we don't even look into it, but the constituents of the chart will be something like shown here. Now let's go to culture media type and protocol. So we were talking about back to nature or sequential culture and let the embryo choose or a single step culture. These are the two basic type of culture protocols available, and we'll also see a comparison between the two. So when we say back to nature, we want to mimic the in vivo environment of the oviduct as much as possible. Because we already saw that pre and post compaction embryos are subjected to a very different kind of an environment. We also saw that how the maternal genome is active and later on the embryonic genome takes over. So, and we also saw pyruvate and glucose preferences. We saw that the embryos initially do not use glucose, they use pyruvate and lactate, and in fact, glucose in the initial culture environment, when we tried to do it in our, from 1920s and all, glucose used to have this two-cell block if you added too much of glucose in your culture media. Then we have role of non-essential amino acids and essential amino acids in the pre-implantation embryos, and inhibitory effect of EDTA on blastulation. So people who developed the sequential culture media saw all of these and tried to implement two different kinds of media. They said that the energy requirement is changing in the pre and post. And glucose in all species does inhibit the two-cell block. So changing the quantity of glucose in the cleavage and blastocyst media is necessary. They said you cannot have one media which will have glucose at a similar level. Since your embryos need a different type of energy substrate initially, have a cleavage media, pre-compaction, and that should be changed or renewed post-compaction by the blastocyst media. They also took care of EDTA, saying that has a detrimental effect on the blastocyst development. So they removed EDTA altogether from the blastulation media. They hypothesized the role of amino acid on pre-implantation embryo, that the cleavage media will contain only non-essential amino acids because the essential amino acids are not needed at that stage, whereas the blastocyst media contains both NEAA. Another important factor which was seen was chemical breakdown of L-glutamine, which is a kind of an amino acid again, that in aqueous solution, when L-glutamine would break down, it would accumulate a lot of ammonia. And this ammonia has detrimental effect on the embryonic growth. So they asked to renew the media, and so that the accumulated ammonia could be gotten rid of. So in sequential culture, as a rule, you renew the media. Sometimes some people do it on daily basis, but not anymore. But you usually renew it on day 3, and that way you get rid of the accumulated ammonia. Now, this is in this debate, single step and MonoStep culture, which was based primarily on the works of Driggers and Communal. They said that let the embryo choose. As long as concentrations of all the things like glucose, amino acids, EDTA, as long as all these concentrations are within tolerable ranges, the embryo itself will adapt to its environment and utilize whatever is needed. And their strongest point that they argued on was the pathophysiological stress. When you switch an embryo from one kind of a media to the other kind of a media on day 1 and day 3, you know, it has a lot of stress. There are osmotic and pH changes, temperature changes. All of this could are stress factors for the embryo. and we'll be depriving the embryo of some autocrine paracrine factors that may have been produced during the first culture period. They also had said that glucose is present in the natural environment in the fallopian tube itself, and there is evidence of culture media with glucose that do not inhibit embryo development. So, say experiments they cultured embryos with glucose, and they found that it does not have any detrimental effect. The role of EDTA. The negative effect of EDTA during blastulation refers to a higher concentration, but the presence of low quantity is not deleterious to the culture of the embryo. Use of mixture with all 21 amino acids. This way, the embryo would be able to choose which one to use by itself. So instead of first giving one kind of amino acid and then the other kind on the cleavage and blastulation media, the single step media provides all the 21 amino acids and lets the embryo choose whichever amino acid it needs. The problem of accumulated ammonium from decomposition of GLN is resolved by using a more stable dipeptide of GLN. These are called glycol L-glutamine or L-alanine L-glutamine. So the accumulation of ammonia was said because of the breakdown of L-glutamine. So if you replace it with a more stable dipeptide, then there will be no accumulation, and single-step media should work. You should not have to renew it just because of the concern of ammonium accumulation. Now, a brief comparison between culture period of commercially. Um, I'm sorry. Uh, Sorry for that interruption. So, comparison between the culture period of commercially available media. You have a one media system and sequential media system as mentioned here. So these are a few companies that provide you single step media and a few companies that provide you sequential media. What is important to note here is the culture period. The culture period of this is something very important that you should know the moment you get an embryo culture. Any new media that you get, you should know from which day you can culture your embryos in it. So like you see, some of them say day 1, some say day 0. So this is the most important part here. That says that you can culture it from day 1. You cannot, just fertilize and oocyte and leave it in. It is a cleaving media where you have to transfer it on day 1 post fertilization, post the 2PN check. While mediums that mention day 0 are the ones where you can simply, do an ICSI or post a long incubation. You can simply keep your embryos in there for extended culture. Now, day 0, day 1, like I mentioned, it is important to note that many media do not recommend culturing oocytes in them. So if you keep your oocytes in them, then it will be detrimental for the oocytes, and they will not perform, it could affect its fertilization later on. Now, stage IVM, if you call about in vitro maturation of oocyte, then stage IVM medium is the only kind of commercially available medium which is meant specifically for in vitro maturation of oocytes. However, studies have shown that you can successfully use blastulation media for the same. So at times, if you get all immature oocytes and you want to attempt you denudate those sites and they are all immature. The only choice that you have left with is probably do an in vitro maturation. At that point, you should use a sage IVM medium or a blastocyst medium. Not be the blastocyst medium may not be as good as the say which is specifically designed for IVM, but it is a good choice in absence of an alternative. These are the kind of protocols that we usually follow in our lab. So in a sequential media, you have an uninterrupted culture till day 3. And after day 3, move it to the blastulation media, if at all you are going for a blastocyst culture. Now, sequential media with a fresh state-specific medium, this no one follows it. I haven't seen any lab that follow it. There are single step, and then the other one is the single step or a MonoStep culture, and the same media is used throughout the 560s. And single step media with an interrupted culture where you do a media renewal on day 3. So there are many labs who go for a single step media because of the ease of protocol, and yet they change the media on day 3. This is their own protocol. Maybe they have seen some differences in blastulation and all. That's why. Now, if you have to compare the protocol, so based on these characteristics, we are trying to compare a single step or a mono-step culture protocol with a single step interrupted or renewed protocol and a sequential with renewed. You see a lot of smiley points and a lot of sad faces. Now, the main factors, which, these are the factors based on which any IVF lab should decide what kind of protocol they want to follow. So, leaving the embryo undisturbed, everybody should ideally do it, right? But, you see that at the cost, it is at a cost of higher blastocyst, lower blastocyst rate and all. Maybe you would decide to go for a sequential media. This protocol varies lab to lab. Replacement of essential nutrients, accumulation of toxins, environmental stresses. I think this is a very important factor. The environmental or the pathophysiological changes because of the actual physical move of the embryo. Then labor intensity of the embryologist, which most of us are very concerned about. Single step uninterrupted culture is the easiest thing to do. So if you have new embryologists, if you have embryologists who are trainee and not that proficient, I think you want to choose this protocol. Again, cost, time management, and QA/QC. All of these factors needs to be considered before you actually decide on a protocol. Single-step media are, less expensive because you don't need 2 kinds of media for a single patient. You just need 1 type of media. So these are the factors that you have to weigh in and then decide which protocol you want to choose. So after going through all this, which protocol is superior? Basically, the studies are completely inconclusive. So if you are doing sequential and it is working for you, you should stick to it. And if you are doing single step and it is giving you an industry, benchmark standard of fertilization and blastulation, then you have no reason to worry or thinking of. After the embryo culture protocol, we come to the embryo culture strategy. Embryo culture strategy is whether you want to culture individual embryo individually or you want to culture them in a group. What the various, and we want to see various advances that has happened in the culture strategies. These are the kind of dishes that every IVF lab uses to culture their embryos. So you have single embryo culture for which you will make droplets and then you will keep embryos in single droplets, or you have specialized dish these days where instead of forming droplets, they have micro wells in which you can keep your individual embryos. And okay, let's briefly see what an individual culture, what are the advantages and disadvantages before we actually choose one type of a strategy. Advantage for an individual evaluation, the first and foremost advantage when you do an individual culture is you can individually evaluate an embryo morphology and its rate of development. This is very important. If you do things in a group, you won't be able to tell. So you won't be able to tell or mark the morphological characteristics over the course of 3 to 5 days of development. You won't be able to do that. So this is the most important advantage of an individual culture. Now, embryos that are cultured together may be exposed to secreted detrimental factors too. So if you have one embryo which is apoptotic, some cells, then if you culture them in a group, then the detrimental effect of uh, the embryo which is is going to apoptosis might affect the embryos which are good grade, and depletion of necessary substrates by other embryos if cultured in a group. So whatever is a negative part of a group culture is advantage. It comes as an advantage of an individual culture. So we say that when you culture them in a group, it also, the embryos could end up depleting the necessary substrates which are present in the culture medium. Now, talking about the disadvantage, the individual culture is again labor and cost intensive. Special kind of plating at times, if you happen to choose that, adds to the cost, and the 15, 16 sites and trying to keep them in different micro droplet could be labor intensive. However, with practice, even practice doing this, then it doesn't seem cumbersome. And loss of autocrine, paracrine growth factors affecting growth. So, embryos like being together. They don't like being alone. So when there are things called autocrine, paracrine factors and growth factors, which every embryo, they share among themselves and it enhances their own growth. So if you culture some embryos individually, you might lose on the autocrine, paracrine and growth factors. pH, temperature and osmolality during the manipulation of this could be a concern because individual droplets have few microliters of embryo culture media. So smaller the droplet or smaller the quantity of media, more difficult it is to maintain its temperature, its osmolality, as well as its pH. If you have a larger amount of the media, it's much easier. So what are the advantages of group culture or communal culture? Let me see where I am on the timeline. Okay. It's an easy protocol. I'll have to be fast, I think. Okay. Group culture or communal culture is easy protocol. You use accumulated autocrine, paracrine growth factors, and you have less exposure to temperature changes during the manipulation. Disadvantage, it does not permit continuous evaluation. This is the biggest disadvantage of group culture. You cannot individually evaluate them using time lapse or or even individual marking individual morphological characteristic is not possible. And the embryos in a group may be exposed to negative effects of a dying or a delayed embryo, which are cultured in the same well. So benefits of group culture are more evident when you plan a blastocyst transfer. So this is important to note. A group culture benefit usually comes into picture only when you are planning a blastocyst transfer or an extended culture. Now these are some advances in the embryo culture platform. I think I'm just mentioning that you have things like rotation, shaking, pivoting, vibration, and piezoelectric actuators. We are not going into detail of it. These are few advances which is the future of the culture platforms which are being looked into. So let's quickly move to temperature, pH, CO2. There are culture conditions. So most important, temperature, pH, CO2, and oxygen tension. So like we had seen in our slide of a pre-compaction and post-compaction embryos, there's a difference between the temperature even within the oviduct at the site of fertilization to the time, where it is implanting in the uterus. So what is important to note here is the minimum temperature may not be as critical as the maximum temperature. There is something called the quiet embryo hypothesis, which says that viable embryos have a quiet metabolism, and cooler temperature, they say, could lead to a quiet metabolism. This is just a hypothesis, by the way, because the randomized control trials are inconclusive. But again, having said that, and even noting the fact that fertilization site is 1 or 2 degrees less as the core body temperature, It is important to note that the minimum temperature may not be as critical as the maximum temperature that we are subjecting our embryos to. Reduction in temperature will only slow down the enzymatic function. But rising temperature, even by say half a degree or degree, mostly affect your oocytes maturation, the spindle formation. The spindle might not form back, and it might affect your fertilization rate and retards further embryonic development. Coming to the pH. So pH is study of pH is just like chemistry. It's actually complete your undergraduate level chemistry. pH in the media. Now again, pH is differential within our oviduct. The site of fertilization has a higher pH. Site of cleavage has lower pH, and blastulation is again higher pH. So we have a high, low, high pH. The acceptable pH range that we culture our embryos into is usually set from 7.2 to 7.4. These are the various factors which affect the direction of an embryo culture media. It is the amount of bicarbonate which is present in the media, the concentration of amino acids which are added to the media. Now, the first 2 points, the amount of bicarbonate and the concentration of amino acid, we can't do anything about it. This is the formulation of the culture media. The culture media is delivered to us. We are not making it, right? So what are the other things that we do that impacts the pH? So how much volume of media are we, sorry, how much volume of the media we are taking? What is the oil overlay? What kind of oil are we using? How much oil are we using? Kind of culture dish that we are using, the altitude of the lab, the CO2 concentration in the incubator, and temperature. This all affects your pH. And like we see, there's an inverse relationship between pH and temperature. So when you rise your temperature, the pH lowers, and when you rise your CO2, the pH again lowers. Now, how do you change pH without actually changing temperature and CO2? Already saw we have incubators where you maintain a particular temperature, you set that value, and you maintain a particular percentage of CO2. Don't change it on a daily basis. Now, having said that, what happens? How do you change pH without changing the temperature? Because we have to be close to the high, low, high of, like we saw fertilization, cleavage, and blastulation. We do this by changing the bicarbonate concentration in culture media, and this is what primarily the sequential culture media group emphasize on. So they have a different bicarbonate concentration in their cleavage media, while they have a different one in their blastulation media. And even keeping the same this media in the same incubator with the same set point of CO2 and temperature, you can achieve different pH for the culture. Now, like how pH measurement of pH ideally should be done every day, but it is very cumbersome. It is easier said than done. The instruments for measuring pH are expensive. So I'm pretty sure that most of the labs, yeah, and even outside India, even in Europe, they don't regularly or every day measure the pH every day. The easiest way of dealing with whether the pH is right or wrong is by giving it a visual indicator. Some culture media, not all though, some culture media give it a phenol red indicator. A phenol red has a property of changing its color depending on the amount of pH. At very low pH, phenol red is almost colorless, while at a higher pH, it has a bright fuchsia color like the one which is shown in this culture media bottle. How do you regulate pH? A buffer is something that is added to keep the pH of a solution constant. These are the two definitions that you need to just briefly understand. Buffering capacity is the resistance to changes in pH when a strong acid or base are added. So HIPIS and MOPS, these are two buffers. So we saw that, in our previous slide, we saw that you can maintain pH. If you have a culture dish with culture media, you keep it at the the incubator. You maintain a temperature and you maintain the CO2 concentration. It will be stable, right? Now, this is fine as long as you're just culturing it without having to take it out. Now, there are certain things like, denudation of an oocyte and even screening your OPCs for that matter, certain kind of gamete handling that you have to do outside the incubator. How do you maintain your pH when the gamete is outside the incubators? So this is where buffers come into picture. So you have this buffer media in which you do all the gamete handling. Now buffer media are the two most successful buffer medias are Hipis buffered media or Hipis does seem to have buffering capacity which is closer to our physiological pH that we need. However, what is important to note here is the moment you take a dish outside an incubator, the temperature is going to fall. It's not going to be 37 degree. And since pH and temperature have an inverse relationship, the moment the pH falls, the buffering capacity, which is 6.93 of MOPS, the pH is going to rise and probably it's going to be somewhere 7.2, between 7.2 and 7.3 that we are targeting. So HIPIs and MOPS buffered medium are also Zwitter-ionic buffer or Good buffer. They are named after the scientist Norman Good. And at times we also, these days we have also seen that instead of using just simple HIPIs and MOPS, you have a combined buffer, which is a mixture of HIPIs and MOPS and sometimes some other kind of buffer. Like for example, we have a 1:1:1 for HIPIs, MOPS and DIPSO. Now quickly coming to the oxygen. First, noting down fact. The fact is the embryo passes through sequential oxygen concentration, which varies from 5% to 2%. It is not constant at all. The optimum oxygen tension of the in vivo embryo it is dependent on the stage of development, whether it is on a pre-compaction stage or post-compaction. And post-compaction embryos benefit more from lower oxygen concentration. This is also a fact. Now, in vitro embryos are subjected to, this was the fact for what happens in our body. Now if we compare it with what we are subjecting to in the lab, then in vitro embryos are subjected to a monophasic oxygen concentration. Why? Because you're keeping them in an incubator, which has a only, it's monophasic. You can give only one particular concentration, either atmospheric or if it's a dry gas, a 5%. Embryo culture at 5% oxygen results in improved morphological parameters, reduced embryo arrest, and higher live birth rate as compared to oxygen, atmospheric oxygen concentration of 20%. There is abundance of data which shows that atmospheric oxygen negatively affects the pre-implantation stage. So these are the two type of incubators that I have tried to show here. This is a box incubator. It looks like a box. And this is a benchtop incubator. So coming to box incubator typically uses an atmospheric oxygen concentration of 20%. So in the room around us, we have 78% nitrogen, 20% oxygen, and 0.04% CO2, right? So the moment, so box incubators will use one kind of a gas, CO2 cylinder, to maintain the CO2 level. However, they will rely on the atmospheric level of oxygen. So the moment you open the door of a box incubator, the atmospheric air will rush in, and in order to, and then the CO2 incubator has sensors in it, so it will automatically reduce the CO2 level. So the sensors will detect the reduction in the CO2 level and appropriately will purge the CO2 gas again back to it so that it comes to a 6% level. And this means that the oxygen level will still stay at close to 20%. Now, the box incubator initially, which was used for somatic cells, were larger units of 150 to 200 liter. However, dishware, which is used for IVF, is considerably smaller, and hence even a 14 to 48 liter box incubator should work for the media equilibration and culture. Important thing to notice, if you want to buy a bigger box incubator for culturing more dishes, for example, or equilibrating more, you have to remember that it also influences the gas equilibration and the recovery time. So it takes more time to recover. Coming to benchtop incubators, benchtop incubator typically use 5% physiological oxygen. They have extremely small chambers of 0.31 to 0.5 liter, and this improves the recovery time a lot. Smaller incubator volumes are beneficial in terms of rate of atmospheric recovery following repeated opening. So box incubators, and when you compare with box incubator, the recovery time of benchtop incubator is fast. Most benchtop incubators will utilize cylinders of pre-mixed gases. So the tri-gas cylinders, which has a pre-mix of 6% CO2, 5% O2, and 9% nitrogen. A modern triple gas incubator, though, has a cost implication. So it is not only the first time cost, which is of consideration, it is also the renewal of these gas cylinders. When you compare with the CO2 cylinder, a tri-gas cylinder is much more expensive. Nowadays, like we saw that the blastocyst in a uterine cavity is subjected to only 2% oxygen. Ultra low concentration of oxygen is being studied upon, but evidence is still inconclusive. So this brings me to the end of my presentation and the take-home message. If in doubt, always stay towards the lower temperature. Say you don't have a temperature meter, you have touched a surface or whatever, you feel the temperature is high. Staying a tad bit on the lower side is better, far better than being on the higher side. Best in a benchtop incubator, 5% oxygen concentration is what our body gives to our embryos in vivo. Let us try to match it instead of showing studies which say that it has no effect on blastulation and all that. I think we should all invest in benchtop incubators, and it should not be something that you should purchase only when later on. On the first go itself, purchase a benchtop incubator. Simple things often work best. So simplify your workflow. Don't have a very complicated workflow, because if you have a complicated workflow, it will be very difficult to replicate it for more number of cases. Reduce gamete handling, touch less, as less as possible. Take the dish out of incubator as less number of times as possible. If your cumulative results are good, the word cumulative here is of uttermost importance. One batch giving you 80% result should not have you on the top of the moon, while at the same time, one batch giving you just a 20% result should not, you should not be depressed. You should just go back to troubleshoot and find out what might have gone wrong. So results should always be looked upon cumulatively, more so over a year, so that you have covered all seasons. Lastly, do not be afraid of a change, but change only one thing at a time, even if it is something as trivial as a falcon tube or one kind of a buffer. So just one thing should be changed at a time. Monitor it for a while, and if you are happy with the result, then go and change the other thing. So this is the end of my presentation, and a few references, which was very interesting. If you guys want, I can share this later with you. Okay. Ma'am, I think I'm done. I'm ready for question and answers.
Over to Rafia for question answer session. Please, Rafia. Unmute.
Sorry. Good afternoon, all. I'm Rafia Sangameshwari. Ma'am, I'll read out few questions for you.
Yes.
So, to start with, how is oxygen, tension, and pH of media is adjusted in single-step media?
How is oxygen tension adjusted in single-step media?
Yes.
You also said pH?
Yes, ma'am. Oxygen, tension, and pH of media.
Okay, so oxygen tension, it depends where you are equilibrating the media, right? So if you have a single step media and you're equilibrating it in a box incubator, for example, right? So your oxygen tension in a box incubator is 20%. So I think if I'm not wrong, it takes approximately 7 to 8 hours to equilibrate your media in a box incubator for the oxygen tension and pH. And it also depends on the kind of protocol that strategy that you're using. If you're using, say, a 4-well, the equilibration time will be different. While you're using a center well, it will be different. If you're a micro droplet, it's a different case altogether. But safely, you can say if you keep it for at least 6 to 8 hours, then the media will be equilibrated for the oxygen tension and the required requisite pH. Does that answer your question? I don't know whoever asked.
Ma'am, can I ask next question?
Yes, please.
Yeah. Can oxygen and carbon dioxide level be altered for culturing?
The again depends which incubator you have, right? So if you theoretically, box incubator, you are only providing CO2, right? You have CO2 cylinder and 6% is your target set value for your CO2. And rest of it takes from atmosphere, which is atmospheric oxygen concentration of 20%. So did you, Rafia, can you just mention, was it oxygen and CO2 both? Was the question both?
Yes, ma'am. Oxygen and CO2 both.
If you want to change the CO2 concentration for whatever reason, experimental reason you will just have to set it to, instead of 6%, you will set it to whatever, say 7%, 8%, incubators let you set that. So when you do that, it is going to use appropriate amount of CO2 from the incubator, and that can be available within your incubator. If you talk about a benchtop incubator, it has a pre-mixed gas. These days, you also get benchtop incubators with mixers, where you have to give it nitrogen, CO2 and O2, 3 different things, and it is going to mix it. Actually, you give 2 things and it mixes it. But most of them us use trigas, which is a pre-mix of 6% CO2, 5% O2, and rest of it is nitrogen. So if you have to change the oxygen and CO2 concentration on the benchtop incubator, you will have to get a different premix. You have to go to the manufacturer, ask them to give a different premix, saying that I want to try 2% oxygen for example. So you have to go to the manufacturer of those cylinders and ask them to give that to you.
Okay, ma'am.
Any more questions, Rafia?
Yes, ma'am. One more question is there. Does culture media influence the mosaicism?
Mosaicism.
Yes,
Yes.
ma'am.
I would really want to pretend and say I exactly understand genetics that well, but trust me, it's a very huge kind of a thing to say. I think, as far as my knowledge is concerned, there is epigenetics is such a topic which scientists को नहीं समझ में आया, मुझे क्या समझ में आएगा? I think it have some impact somewhere, but mosaicism is again something some embryos will, more than culture media, I think it is the culture strategy which could, but I'm not very sure on this. I'll have to go find out papers which actually say whether it actually can lead to, some cells being chromosomally normal, mosaicism, while some being, aneuploid, while the some being euploid. In general, an embryo itself, a blastocyst itself, has a lot of mosaics. Not all, you are just, who has taken every cell from a trophectoderm of an embryo and and ran it on a next generation sequencing to see if all of them were euploid or not. None of us have done it. What we have done so far is just taken 8 to 10 cells from a trophectoderm and considered that as the best representation of our embryo and run it through, PGT and seen whether they are euploid or not. And mosaicism, I'm not very sure if a culture media can actually impact it. More so of ICSI or not ICSI and things like that might be more questionable in that.
Okay, ma'am. Ma'am, one more question.
Yeah.
What is exactly mechanism of low oxygen use for embryo culture?
What is the exact what? Sorry?
What is the exact mechanism of low oxygen use for embryo culture?
mechanism. I think it is more to do with how the embryo, the glycolic metabolism takes over. So whoever asked this question, I think Rafia, you would know that who asked the question. उसको पूछो कि कितने अच्छे से अपनी theory पढ़ी है. I think it is more to do with the glycolic metabolism, and within that, I am not very proficient with the cell, the chemistry of it, but lower concentration of oxygen will give you it is a kind of